Hello, welcome to this course of Principles of Vegetable Breeding, offered in the first semester of first year, 2020 to 23. Uh, let us just go through the schedule of lectures just for the entire semester. First 2 weeks, you are going to learn on importance, history, and evolutionary aspects of vegetable breeding, and if there is any variations from cereal crop breeding, definitions of terminologies and genetics and plant breeding. Then the third week, centers of origin and their role in crop improvement, germplasm introduction, conservation, exploitation, and plant introduction. Uh, breeding, the fourth and fifth week, you're going to learn on breeding systems and methods, mode of pollination in vegetable crops, cytogamous, allogamous, and often cross-pollinated vegetables, factors responsible for self and cross-pollination, flowering habits and sex forms in vegetables, self-incompatibility, systems, and male solidity, which is prevailing in vegetable crops. So sixth to eighth week, you're going to learn on methods of pond breeding. That is, methods of breeding for self and cross-pollinated vegetables, selection procedures, pure line selection in vegetable crops, breeding methods to handle segregating generations, pedigree and bulk method of breeding, backcross breeding, and modifications to improve the vegetable crops. Then you are going to learn on population improvement methods. That is mass selection and modifications and other population improvement methods like recurrent selection or different schemes for improvement of vegetable crops. Then from 12th to 14th week, you are going to learn on heterosis breeding, types, mechanism, and basis of heterosis, steps involved in heterosis breeding, economic F1 seed production, this is very important. It's systems prevailing and facilities, which can be facilitated by the prevailing mechanisms like male sterility, self-incompatibility, and sex forms. Then 15th and 16th week, you are going to learn in mutation and ploidy breeding, which is they are being used for improvement of vegetable crops, and of course, especially for the asexually propagated vegetable crops and vegetables suited for a protected environment. 17th to 19th week, you're going to learn breeding for biotic stress, diseases like biotic stress means so diseases, insect pests, and nematodes. Abiotic stress, the temperature, moisture, and salt stress, and then resistance and quality improvement. Biotic and abiotic stress resistance and quality improvement, breeding for water use efficiency and nutrient use efficiency. That's ultimately how varieties can be, selected. Phenotype breeding, varietal release procedure, dust testing in vegetable crops, so they are important, like a distinctiveness, uniformity, and stability of the varieties which are going to release. How that is being done. Application of in vitro and molecular technique in vegetable improvement. That is what we are going to learn as far as theory is concerned. Then the practical part, that is first 2 weeks, floral biology and pollination behavior of different vegetables. Then third to seventh week, you can spend time in the field for techniques, learning techniques, practicing techniques of selfing and crossing of different vegetables like cold crops, whatever the crops are existing in the field, okra, cucumber, tomato, eggplant, hot pepper, etc. In eighth week, breeding systems and handling of filial generations of different vegetables like demonstration of genetic segregation with marker stocks. Any postgraduate students working on this aspect, if the material is there in the field, we can just go through that. And then the ninth week, identification of male sterile and self-incompatible plants. All this depends on the availability of the experiments which are being conducted by these students. Then demonstration of hybrid vigor and working out heterosis. Definitely, many students are working on the cytosis aspect and will get that opportunity. how that hybrid you can see. 10th and 11th week, 12th and 13th week, screening techniques for tolerance for resistance to biotic and abiotic stresses. 14th to 16th week, you're going to learn working out biometrical parameters, standard deviation, coefficient of variation, product movement correlation coefficient, the regression coefficient, chi-square test for heterogeneity, fitness of test for expected genetic ratios, and detection of linkage. Then 17th to 19th, working out biometrical parameters, genotypic variance, phenotypic variance, GCV, phenotypic coefficient of variation, phenotypic coefficient of variation, heritability, genetic advance, or gain. So then you'll have the practical exam. You can also have the visit to the one of the station, which is relevant for this particular course, grading station, involved in any of the vegetables. Then a scheme of evaluation of attendance 5 marks, quiz 20 marks, midterm 25, practical exam 10, practical record 5, assignments 5 marks, and then final exam 30, 100 marks evaluation. And these are some of the suggested references you can refer, knowing like Allard's principles of plant breeding, Gardner principles of genetics, Hayes and workers like Amer Smith methods of plant breeding, also plant breeding principles and prospects. Then Kalu's vegetable breeding, Cole's genome mapping and molecular breeding in plants, vegetable, and plants and vegetables. Then Peter, KV, and Pradeep Kumar, book written Genetics and Breeding of Vegetables. Friends and news are definitely this important if you are very much advanced individual crops breeding to know vegetables 1 and vegetables 2. Then course vegetable cross breeding written first to one chapter for the principles, and then it is individual cross breeding is dealt, but at least the first chapter you can whatever. Then Simmons, Principles of Crop Improvement, and then Singh, Brady Singh, book, common, very popular, plant breeding principles and methods. Then Singh, Ram J, the genetic resources, chromosome engineering and crop improvement, vegetable crops. So yeah, that's what the background of this course. Let us just get into the first chapter, that the importance and history of vegetable breeding. Vegetable breeding is very important because the vegetable crops as such, they are important. Vegetables are important crops. They provide the nutritional security and the economic sustainability, and they are having high productivity. So they are crops of importance, present and also future. And if you just look at the, if we say that the productivity is very high, to what extent you can just compare with other cereals, pulses, and vegetable crops. Per hectare productivity average in India, wheat you have got, this is quintals per hectare. It's given 38 quintals, paddy 26 quintals, maize 17 quintals. That is red gram 7, 8 quintals, Bengal gram around 8 quintals. And then you just look at this potato, I mean the vegetable crops. Potato, they are only 30, 40, even single digit productivity having crops are there like red gram and Bengal gram, pulses. Whereas you look at these vegetables like potato, 193 quintals per hectare they can give. Onion, 151 quintals. Tomato, 179 quintals. Brinjal, 170 quintals per hectare. Tapioca, 335. Cabbage, 222. Cauliflower, 182 quintals. So there is a big difference in productivity That's why they can in, increase the income from the per unit area compared to the cereals and pulses. Of course, they are nutritionally important crop. What these crops also, vitamins and minerals, they'll add, and the national economy, they'll contribute as the horticulture crops are contributing more towards the national agriculture GDP. So definitely they are important, and breeding of these crops is very important. If you just look at the major vegetable producing countries in the world, highest, the number one is China, maximum area and also production. India stands second, uh, and then United States. They'll get Turkey, Iran, Egypt, Russian Federation, Mexico, Spain, Italy. So other countries, no, very very. But if you look at the productivity, we have a lot of scope is there. Countries like the Spain is having 39 uh, tons per hectare productivity, whereas we are having just 17.6 tons per hectare all for all the vegetables. And of course, China is also lacking 23. But the reason for the other countries having so high productivity is the protected cultivation. So there the productivity is very, very high. So there is scope. Means in India, increasing the productivity, there is a scope. And if you just look at the over the years, how the total production has changed, and also the productivity area, it's marginally increased from 91, 92. So 5.5 million hectares to 9.2 million hectares. Whereas production, it is 58 to 58 million tons to 162 million tons in 2012-13, and the productivity from 10 ton per hectare to 17 tons it has increased the scope. And the latest data, if you take a slight uh, improvement, is there, no doubt in that. Um, So if you look at all the horticulture crops, the vegetable crops almost contribute 60% in case of the production, right, among the horticulture crops. But the area, if you look at vegetables, they say 38%. Fruits, they say 29%, but the production share is 31%. So it depends on the productivity. Some other crops you have less, but money value, if you look at even those low production share crops also can contribute uh, for national economy. So it's not exaggeration, but definitely vegetables are having maximum production contribution among the horticulture crops. Uh, but the area share is 38%, whereas production share is 60%. Which are the important crops? If you just look at it, these figures can slightly vary if you take the latest figures, but doesn't matter. But the ranking almost is similar. Potato number one, right? Highest production share. Second is tomato. Third is onion. Fourth is brinjal. This tapioca, then cabbage, cauliflower, okra, peas, sweet potato, and others. Plenty of other vegetables are there, but these are important. Onion, potatoes. So it's potato, tomato, onion, brinjal. And then you have tapioca, some Kerala and other states only. And but yeah, cabbage and cauliflower comes, and okra, peas, sweet potato. Then all other crops, remaining crops are having less than, you can say less than 1%. Leading vegetable producing states in terms of production, again, a slight variation. The West Bengal is number one, followed by Uttar Pradesh, then Bihar, then Madhya Pradesh, Andhra Pradesh, Gujarat, Odisha, Maharashtra, Tamil Nadu, Karnataka. These figures slightly vary, but still you get some idea how that's leading vegetable producing states in terms of area. If you just look at, that it is varying because of the slight variation in the productivity. Okay, these are again these crops: West Bengal, Uttar Pradesh, Bihar, Madhya Pradesh, Andhra, Gujarat, Odisha, Maharashtra, Chhattisgarh, and Karnataka. Right. Okay, this is what there are important wisdoms or vegetable crops are important even among the agriculture crops and then horticulture crops. What is the scope of this plant breeding or breeding crop improvement there for these wisdoms? So first is the yield can be elevated, can be increased. Increased. It can be increased by this crop breeding. If you want to increase the yield, you can do it by proper management of soil and crops involving suitable agronomy and harnessing physical resources. That is one way. And the other way is by using the high potential crop varieties created by appropriate genetic manipulation of crops. That means if you adopt those improved varieties, so that is going to contribute far because any amount of resources you provide, it is being restricted by that genetic potentiality for yield. Yield you can improve by yes this and then the enhance the stability and adaptability. The changing environments, or you release one variety, or you grow the crop here and some other places the production is very the place to place and also from season to season. So so these are the two requirements for the as crop improvement. So, and it's not only just yield, but it is also yield stability, higher yield, stability, that is sustainability, because that's possible only by the adaptability of the varieties. And of course, quality and other things. Any quality is also one more aspect. But if you say resistance to biotic and abiotic stress, that is going to contribute for the yield. So that's why it is covered. So that's the scope. But if you just look at the contribution of human welfare made by the superior varieties is the most satisfying of all methods of increasing production because this adds nothing to the cost of production and also to the pollution, but adds to yield. Of course, you can say that the cost of hybrids is slightly higher, but that's nothing. Just compare the productivity of those varieties. So that means ultimately the cost of production, especially if you have got disease resistance or some resistant varieties, cost is rather going to be reduced rather. So we are saying that in large, it's nothing you are adding to the cost of production, but definitely you're not adding anything to the pollution. You're not using the chemicals, but the fertilizers, it is required. You use organic or inorganic, that's required. So, history of crop breeding, wisdom breeding, just okay. So, history of crop breeding, we can just throw some light on that. And wherever vegetable crops, how they are relevant aspects, changing the in the field of crop breeding. So it may not be always with vegetable crops. Wherever we get some examples. It's started, the crop breeding. If you say prehistoric, also man himself is involved in selecting the best plants for his use. So 1682, Nehemiah Grieve, he postulated that the stamen is male and pistil is female in plants. So that means some understanding that how these parts are. There is male, there is a female part in the plant, and that's relevant for breeding. Then 1694, Or Camerarius, he rediscovered the sex in plants and described sexual reproduction. Oh, this male and female parts are there and they are involved in the reproduction. Then 1716, Cotton Mather, he described the first natural plant and grow can cross. Naturally there is a cross, there is crossing, and we are getting the new genotype, which is a product of natural hybridization. Then 1717, Thomas Fairchild, the first plant hybrid was artificial, but that was naturally having a water method observed. But now the artificially can we do that? The first plant hybrid was artificially produced, and this hybrid was called the Fairchild's mule. So you know the mule. What is that? Having characteristics of both the parents. So he, crossed Sweet Williams and Carnation, that is Dianthus barbatus into Dianthus caryophyllus. So artificially crossed. We got a hybrid here, artificially produced. That's the understanding that how the sex is involved, sexual parts are involved in reproduction, and you can artificially also uh, deliberately you can make the crosses and get something. It's very novel, rather useful for the human beings. 1760 to 1766, Joseph Colrater, the scientist, made systematic studies on artificial plant hybridization and made observations on hybrid vigor in plant hybrid. So he observed systematically, he made the crosses. And then he started observing, oh, there is some change in the progenies over their parents. So observations on hybrid vigor, few plants, hybrids. 1759 to 1835, Thomas Andrew Knight used hybridization as a technique for plant improvement. He observed that male and female sexes make equal contribution to the first filial generation, and segregation takes place in F2. So this understanding in the 18th century itself, Thomas Andrew Knight, he used this technique of hybridization, and there is contribution from male and female. So the progeny is going to have the characteristics of both the parents. 1856, Louis D. Wilmorin developed a progeny test and employed it to improve the sugar content of sugar beet. It is also referred as Wilmorin principle, right? Progeny test. So what is that? What is progeny test? You know, uh, the worthiness of the progeny is, I mean, the parents, worthiness of the parents is dependent on the performance of the progeny, not its own performance. So that is progeny test, right? You test for a particular individual. If that individual can give a good progeny, then only the value is great. So that is called, the principle, Wilmorin principle, progeny test, right? So you decide the value of the parent based on the performance of its progenies. That's very important. What it is now is not important, but what it is going to give in terms of progenies is important. Progeny test. Okay. So worthiness of the plant or individual is dependent on the performance of the progeny. So that's the progeny test, Wilmore principle. Then 1888, Strasburger described meiosis in what is that actually sexual reproduction you are saying, and both parents are contributing. How it is happening, the very basis to get into this meiosis, right? This is involved only in case of sexual reproduction. Asexual reproduction, only mitosis is involved. So meiosis, how separation of the chromosomes or crossing over of the chromosomes, all is taking place. Right, that understanding is very much essential uh, for the reproduction, and of course, ultimately from the improvement of language. 1877, it's not only just plants, even in animals also, any living things. This is going to be applicable. 1877, Darwin published the books on cross and self-fertilization in Vegetable Kingdom. So there is, because they are bisexual, plants are bisexual, majority of the crops are bisexual. Uh, within the plant also, pollination can happen, fertilization can happen, that is self-fertilization, or even although the bisexuality or uh, unisexuality is, if it is there is a possibility that unisexuality, definitely there will be more chances of cross-pollination. And even in bisexual plants also, there is the possibility of crossing from one plant to other plant, pollen from one plant. Uh, falling onto the stigmatic surface of the other plant and leading to the cross pollination, cross fertilization. So he published a book on that, cross and cell fertilization, vegetable kingdom. Title of the book, Darwin, evolution, how evolution, a lot of uh, developments to understand how the changes are taking place in any organism, including the plants. 1890 to 1900, Hall Nelson emphasized the principles of individual uh, on interest. Nielsen in 1890 to 1900 emphasized the principles of individual plant selection to establish varieties of self-pollinated crops. So, what is that understanding derived? 1890 to 1900 by Nielsen, that you have to select the individual plants, not just in the bulk. Individual plants, you make selection. That means the good plants you are going to select, high yielding or whatever the characters you are looking at, and that will help in improvement. Individual plant selection is important for to establish the varieties of the new varieties you want to develop. That is very much required rather than simply bulk and do it. Individual plant selection. They started thinking about good plants you have to select and then or seeds of only those good plants you have to carry forward. That's the message, understanding they got it. 1899, Hopkins developed the concept of year-to-row selection method in cross-pollinated crops, now referred as a line breeding. It's method of breeding you refer. But the thing is, the one plant, what is that actually? This is again progeny test kind of symbol here also. Uh, one ear head, right? That is sorghum or any the cereals, one ear head. That means one plant seeds. It has to be sown in a one row, next generation, right? That is year to row. You cannot mix and sow the seeds. One ear head, one plant seeds you are sowing in the row. That means all those are at one place. The performance of that row is going to be a decisive factor for selection of that plant which you have collected at the head, right? Or so rather than you collect all the plants, ear heads, and extract the seeds and sow in bulk. No, individual plant selection, Nielsen emphasized, right? That is very important for self-pollinated crops. Whereas even in cross-pollinated crops also, year to row method is very much required. So rather than simply bulking, that means you are not immediately selecting the plant. You are waiting for your selection because from the year ahead, the whatever seeds you are collecting, you are raising in the next generation, next season, rather, and all those plants, if they are doing well, average performance of all those plants in a row, it will determine whether the plant which you have made selection in the previous season, it's valid or not. It's a tentative. No, visually you are doing it, but visual sometimes it may not be the actual potentiality of that plant. So that progeny is how they are performing. So that's the principle year to row selection. That's what now you call the line breeding, right? This is line breeding. So it's very much important for the cross-pollinated crops. 1864, John Gregor Mendel, all of you are quite aware about this. Mendel's experiments on pea. So he published Laws of Inheritance in 1866 itself. So it was the first authoritative interpretation of the simple facts of inheritance. That is, there is a factor which inherits from the parents to progenies. The first authoritative right interpretation. Otherwise, it's just writing. The knowledge, what is the idea you got, you are putting it in words. But here, it is an authoritative interpretation, the fact of inheritance, right? There are factors which are getting inherited from parents to progeny. The paper Experiments Related to Plant Hybridization appeared in Obscure Journal. It's not a very popular journal, Obscure Journal in 1866, and it was ignored for 34 years because he's not a big man, and the journal is also not very popular. But he found out the very, the course-changing kind of discovery, you can say, or facts he has in the net or brought out and highlighted, that is inheritance. Okay, so he got the Nobel Prize after his death. So when it happened in 1900, three different scientists at different places: Carl Franz Joseph Correns, Hugo de Vries, Eric von Serac. Three, Carl, like that he discovered the Carl Franz, German botanist, and Hugo de Vries, Holland, the Dutch biologist, and the Eric von Serac, Austrian botanist. So all independently carried out the experiment. They have confirmed that whatever the results published by Mandel in that obscure journal are true, are the facts. And yes, this opened a new vista to plant breeders with, you know, a completely strong background that there is factor which is inheriting from progenies, I mean, the parents to progenies. So now, what is it actually? How this is important? Uh, you know, uh, Plant breeding. Yeah, there is again you have two different, the concepts which are very much dependent: genetics and breeding. So genetic background. If you have, your principles are strong. They are true, right? If you, that's what the genetics. You actually have the knowledge of genetics. Then only you will be a good plant breeders. Without knowing the genetics, how the genes are inherited and all, without that, your plant breeding is, you don't know the very basis. It's very fundamental of your plant breeding is genetics. So you should know the genetics. Then only you can become a good plant breeder. Sometimes, some poor genetics also, they say it's very good plant breeders because you may be lucky. You're not a good thinker. Some good variety, by chance you got it, but without applying the knowledge, you may succeed. But the thing is, with the application of the science, that is the knowledge of genetics, if you achieve, or if you are achieving, then you are really, it's not the fact of whether you are lucky or not. Lucky means you just get into the field and some good plant you encounter, you will be lucky. But you are having the systematic way of searching that plant, or creating opportunities for uh, the getting such plants. That is possible only when you are a good genetist. You have the knowledge of genetics. So that is what the basis for this. That's whatever the laws of inheritance are there. The genes are there. Genes are getting inherited to next progenies. And what is the rule that they are inherited, how they are inherited, and all that is there. And how, within the gene, you have got two alleles, the one dominant and one recessive. All that is going to uh, be very, very important for the plant breeders to achieve his goals. So then the 1903, Johnson, he proposed the concept of pure line theory on the basis of his studies with French bean, vegetable crop, right here also. And that Gregor Mendel also studied with the vegetable crops, peas, garden peas, right? All the seven characters or the eight characters, whatever, had chosen. That is with the peas, and this is with the French bean. That means, it's really great that vegetables also have a relevance of such important developments in plant breeding. He proposed the concept of pure line theory on the basis of his studies with the French bean to improve the seed size. He was the first man to give the name gene to Mendelian factor. There is factor you are calling. What is that? Its factor is inherited from parents to progeny. That is the terminology gene coined by Johnson. 1904 to 1911, G.S. Tell and Dr. Edward East put forth the concept of heterosis. Now, today, the world is being fed by these hybrids. This is the concept where Sell and Edward put forth. The 1918, Dr. Donald Jones he proposed the double cross concept. Double cross concept was suggested during 1921. The first maize hybrid seeds were listed among, given to the farmers. So practically, rather than, not just in the experimental fields, it was distributed among the farmers. That's very important. So what is that double cross hybrid? Why, the hybrids, heterosis is there. Heterosis is there, but why this double cross concept? Two parents, if you cross, you will get a single cross, right? Such single cross, two different single cross. If you cross, you will get the double cross, right? Why these four parents are involved? What is the advantage? What is the disadvantage? Why this is successful in maize? All that is going to be there. The maize being the cross-pollinated crop, right? You have got the male part and female part. Even sexuality is there, but at the same plant, you have got male and female parts. So this, some relevance on the uh, presence of heterosis is one, and also for getting heterosis, you have to select the parents. I mean, you have to develop the parents lines, pure lines you have to develop, right? In cross-pollinated crops, you may not, the completely call that as the like homozygous line, but maximum amount of homozygosity is there. So you have to develop the pure lines to get the hybrids, right? You have to cross two parents, then only you'll get the hybrid. So that homozygosity, you're getting it. When you are getting the homozygosity, there is a possibility of inbreeding depression. Inbreeding depression because the lethal alleles are there in heterozygous condition. They are not being allowed. What is that one? Dominant allele. The another is recessive allele. The recessive allele is detrimental. That means the individual carrying that allele is going to die or is going to not perform well, right? That's what detrimentally means. But that is there in the plant system because of heterozygosity. Because dominant allele, which is safe, which is masking the ill effects of the recessive allele, and it is preserved and it is, but it is not allowed to express. So if you self, that will have the homozygous chances of getting into homozygosity that will express, and that is called impeding depression, right? You get the individuals which are carrying the lethal alleles which are brought into homozygous, their performance is poor. It's not just dead; it is any kind of reproduction or any performance of plant if it is negative, right? That's impeding depression. So that is what you are getting into this at the time of development of pure lines for getting your hybrids. So what to do that? So you will have the poor hybrid seed, which you have to give it to the farmers. For that reason, two single crosses are crossed to get a double cross. You are foregoing or you are sacrificing the uniformity. Two single crosses, if you cross, you will get the heterozygosity, right? The two hybrids, right? If you cross, that's more amount of heterogeneity. That plants are not homogeneous. right? So that's a negative point, but still you can't help it to avoid that inbreeding depression for seed yield. So that's why double cross concept was proposed, and practically it was uh, possible to distribute the seeds of double cross hybrids to farmers. That is Jones, right? Donald F. Jones, what he has done. So these are the things. If you know the complete concept of this genetics, the plant breeding becomes, handling crop breeding um, concept is easier. 1919, Hayes and Garber suggested the idea of recurrent selection first time, and same independently by East and Jones 1920. We have what many varieties. This recurrent selections. Recurrent selection, you can get directly the varieties, or you can also get the lines for hybrid breeding, right? It depends on the type of recurrent selection you are going to adopt. So that's Hayes and Garber. Very important. East and Jones, 1920, next year. 1921, Muller observed the mutagenic effects of X-rays on the insect Drosophila. So there is one more concept, how the changes are there in the individual. Some changes are brought in. That is through mutation. What are some agents are there which can alter the chromosome, which can alter their DNA. So by altering or disturbing the chemical structure of that DNA, you'll get the changes in the genes, and the morphological changes are also going to be there. So this X-ray is going to have that effect, right? The mutagenic effects of X-rays in the insect Drosophila observed. So that means, so there are some agents which can bring the change mutations. 1928, Stadler, application of X-rays to increase the rate of natural mutation was realized in barley. So now this, this has got a mutagenic effect, and why can't we use it for increasing the rates of mutations? So if it is for the good cause, so that possibility was brought out by Stadler in 1920. 1933, Rosales discovered the phenomenon of male sterility in maize. Oh, there is male sterility in maize, which is going to be now can be used for economic way of seed production, because for developing hybrid seeds you have to avoid selfing. To avoid selfing, maize sterility is going to be useful. So in that sense, so this is important. So Rosales discovered the phenomenon of maize sterility in maize, and Davis suggested the use of top crossing uh, for initial screening in maize. For initial screening in, okay, top crossing. What is that actually? What is the meaning? You're going to learn some of the terminologies. What's that top cross? You're going to um, use it, right? To know some amount. I mean, certain concept like combining ability and all that, you can do it. So it's very much required for selection of the lines in the crop, especially cross-pollinated crop like maize. So Davis brought out that, and then 1900 successful attempts were made for production of seeds of temperate vegetables in Quetta in Pakistan, because Britishers were ruling us. A lot of temperate vegetables they introduced. We are habituated, but the seed production of that is being done in a temperate region. That was now the Quetta. Now it is in Pakistan. They did that. So that means it's a kind of crop improvement only, kind of introduction, and then producing the seeds of that. You cannot produce it in our tropical region, those temperate vegetables. 1942, Tisdale and co-workers suggested polycross for screening purpose. So polycross, what do you mean with polycross? Polycross, right? Polycross, can you make several crosses on the same plant? Polycross is not done artificially, remember. It is one plant at the center, surrounded by different, genetically different plants, and collection of the seeds from that single center plant, which is, some few flowers are crossed by plant A, few flowers are crossed by plant B. Like that, all surrounding plants are going to contribute in the form of pollen. And if you look at the different fruits or the seeds, they are derived from different plants. So in a bulk only, not don't think that one seed will have a contribution from more than two plants. Female is fixed, that one plant, and then polycross means allowing it for open pollination. Cross-pollinated crop it has to be, right? You are avoiding this. If you want to avoid, right? Otherwise, also it's a cross-pollinated crop naturally happening. You collect the seeds of that. That is referred as a polycross, right? This is required. Suggested for polycross for screening. It is the like coworker. Then 1943, Jones and Clark. Yeah, you know the application made solidity is there, but how to use this? It took a long time to utilize that. Oh, this is going to be useful for hybrid seed production. Avoid the emasculation process. You have to emasculate, right? Otherwise, it will be self-pollinated naturally. So avoid that. You have to emasculate. That is being done naturally by usage of maize steroid lines. So Jones and Clark demonstrated application of male solidity for exploitation of hybrid vigor in onion. This is again one more milestone happened with respect to vegetables. 1945, Dr. Hull suggested the use of recurrent selection for specific combining ability. Use of recurrent selection for specific. Now several schemes came in recurrent selection. You can improve the specific combinability. You can improve the general combining ability, or else simply OP variety also you can develop. So depending on the screen. So Hull suggested for the use of this recurrent selection for specific combining. It's the only kind of the mating parent you are going to use that will improve the specific combining ability. When once we start learning on the recurrent selection, we'll come to know what is that, how you can improve. So, but Dr. Hull suggested this for improving specific combining ability. So, hybrid breeding is very important. Heterosis breeding. Yeah, this is what there's some milestones and what has happened in the vegetable research at IRI New Delhi or ICR system. That was a important organization which is working on vegetable crops to help pollination. Improvement work on vegetables started at IRI New Delhi with the establishment of small section of vegetables in the erstwhile division of botany in the year 1940, which later became the part of division of horticulture after its establishment in 1956. 1940, division of botany, then horticulture division. Okay, division of horticulture. After that also vegetable improvement was there, which is the main objective of to conduct research in all the major horticulture crops, including vegetables, and to import. It's also to impart the postgraduate teaching. Then the research work was strengthened further with the creation of Division of Vegetable Crops and Floriculture in 1970. 1982, floriculture was separated and the Division of Vegetable Crops started functioning independently. The division was renamed as Vegetable Science in 2004. The mandate of the divisions when they created to conduct applied and strategic research and improvement of vegetable crops, to impart the postgraduate education and training for human resource development, to disseminate information on recent advances in vegetable crops, to provide advisory and consultancy services on crop and seed production technology, and the improvement of vegetable crops, to collect, evaluate, purify, preserve the biodiversity material related to different vegetable crops. So that is very important in collaboration with the NBPGR, separate organization for collection of germplasm, and utilize the desirable superior genotypes in the breeding program. So vegetable research in India got momentum in 50s, 1947 after independence. The nucleus plant nutrition scheme was introduced at IRI immediately after independence, and that has resulted in selection of 30 good varieties. Dr. Harbhajan Singh had division of plant introduction, released some of the varieties like Pusa Savani, Bendy, Pusa Purple Long, Pusa Purple Round, Pusa Purple Clustering, Brinjal, so many. Then 1949 established vegetable breeding station at Katrain in Kulu because we got separated. Pakistan was created. Kata is not in our hand, but somewhere we have to have our own facilities to produce the separate vegetable crop seeds. So they established the vegetable breeding station at Katrain in Himachal Pradesh to produce the seeds of temperate vegetables. Now also it's the regional station of IRI. In 1955, this institute was transferred under IR, right? So then 1971, ICR started AIC, All India Coordinated Vegetable Improvement Project at IRI, New Delhi, with 17 centers, multidisciplinary research for improvement of vegetables, headed by project coordinator Dr. Vishnu Sarup. He was the first project directorate or uh, project coordinator, rather. Project coordinator. So 1971. So that AICVIP now also is existing. It is just having the network of collecting the almost developed varieties and testing them at different places, and then giving the feedback and ultimately finally helping it to release the variety. Multi-location trials. You are a plant breeder. You want to have the multi-location trial. It's very difficult, but you can involve in this network. There's AICVIP. You can give your material. You also can conduct. So you'll get the material. Your material is being tested at different places of your interest, or whatever it is decided in the workshop annually held. So 1986-87, it was upgraded as the project directorate, right? It is a project coordinator, yes, project coordinator was there. AICP only was there, but center AICP only was there. But now, I mean, 1986-87, it was upgraded as Project Directorate of Vegetables Research, and first project director was G. Kalu, right? Vishnu Sarup was the first coordinator, but project directorate. So, in addition to just testing, some amount of research work also being done as a project director. Just like now also you have got like Puttur, you have got cashew, and onion, you have got project directorate. So like this, giving importance, giving more funds, giving more scientists, not not just test the varieties or give it to someone else coordinating the testing of the varieties throughout the country. It is you can have your own. That's what the project directorate means. 1992 headquarters was transferred from New Delhi to Varanasi, right? That project directorate. And 1919, 2000, it was upgraded as Indian Institute of Vegetable Research. That means project director means it's a low profile. Now, if you upgrade, it will be, the more resources, more scientific manpower, more funds, more intensive research you can carry out on more number of vegetables. That's what this Indian Institute of Vegetable. That same place, Yagat, that AICRP project coordinator also. Director is one post, and then AICVP project coordinator is one more post. Two different personalities are there. The AICRP project coordinator is only coordinating the trials being carried out throughout the country, whereas the director of Indian Institute of Vegetable Research having his own research activity in different vegetable crops, having different divisions, right? Like you have seen, must have seen the IIHR, you got vegetable, you got foods. There, you may be having different crop improvement, crop production divisions, which are exclusively working with vegetable crops. 2005, 2006, there were 8 main and 18 sub-centers and 22 volunteer centers. IIVR, IARI New Delhi, IARI Katrain, IIHR, then TNA Coimbatore, PIU Ludhiana, Rahuri, Sabor. These main centers were there for this AIC VIP. Okay, then volunteers, 18 sub-centers. The importance is more like main centers and 8 sub-centers. Then voluntary means ICR is not providing the funds there. The individual institute or universities are participating there. Once their role is confirmed, they are doing well, they can be taken as a sub-centers like that. If the role is much higher, then it can be made as a main center. Okay, this is AICVIP's way, how the institutes are, the centers are there. Even just that means 8 plus 18, 26 plus 22, that's like 48 odd centers the vegetable varieties are being tested. For varieties, crop improvement, of course, agronomical trials also they carry out. It's not just your breeding trials. 1956, creation of separate division of horticulture at IRI. Do we collect that? Horticulture division, already said. 1960, establishing SAUs. It's not only just ICR we have discussed now. State agriculture universities. 1960, GB Pant University of Agriculture contributed a lot in vegetable improvement. 1962, Rajasthan Agriculture University, Orissa University of Agriculture Technology. Then Punjab Agriculture University, 1963. They contributed a lot in vegetable improvement. Then in 35 years, 342 varieties in 22 different vegetable crops, which 219, 209 are open pollinated and 93 are hybrids and 40 are resistant to disease. This is what the contribution by the ICR and also state agriculture universities which are involved in vegetable improvement. And this has resulted in development of varieties disease and pest resistant one like Pusa, Savani, Okra, YBAB, Arka, Anamika, Okra, YBAB resistant, when Pusa, Jwala, chili, virus and aphid resistant, Pusa, 120 tomato, root knot nematode resistant variety, Pusa, Samrat, Brinjal, Pomopsis, blight and shoot and fruit borer resistant variety, Panth Rithras, Brinjal, field resistant to bacterial wilt, then Arka, Rajhams, muskmelon, powder mildew resistant variety, Arka Manik, multiple disease resistant varieties like downy mildew, powder mildew, anthracnose, like that. Many varieties. Few are, just mentioned here. Plenty of contributions with respect to, that is, some 20, 10, 15 years back. That now, if you take it, that number will increase, right? Plenty, much more. So areas of major impacts of vegetable breeding. What has happened? Hybrid seed production system becomes easy by self-incompatibility in cold crops and gynesis lines in the cauliflower rather. Gynoecious lines in cucumber and muskmelon, the use of technique, self-incompatibility, or for cold crops like cauliflower and gynoecious lines in cucumber and muskmelon. Hybrid seed production is a tricky, right? You have to go for the crossing. That is going to be, the intensive. They were intensive one. Then identification of N53 onion variety and development of production technology has led to two-season onion production in North India. Otherwise, the Kharif onions were not there in North India because of all this. Even during Kharif also, because now also you see the requirement of North India in the Kharif end. It is from the south, right? So you have a problem of growing particular few crops. Now, because of these developments, you can grow any vegetable anywhere. Then at least some difficulties will be there, but because of these resistant varieties or the varieties adapted to the condition, led to growing those varieties in different places and in most of the seasons. And then cauliflower development of Pusa early synthetic made possible to grow cauliflower in warm South India. Synthetic varieties are having higher temperature tolerance. Otherwise, these are cold crops. Then India has almost achieved a self-sufficiency and started exporting these vegetables. So that is the major contribution you can say. Again, these scientists Camerorius, Otto Kohlreuter, Mendel's, Norman Borlaug, the Compton Crick, and Adson and Crick, the involved in Borlaug, Nobel Prize, Mendel Nobel Prize. Some of these scientists photographs can test. Yeah, this is what the chromosome, DNA, nucleic acid that contains all the genetic instructions used in development and functioning of the known living organism. When what phase it unfolds, and then it contracts and it just situated in the nucleus. As understanding, and plants account for 80% of the human diet. That is, 30,000 terrestrial plants are known to be edible. 7,000 are cultivated or collected by humans for food. 30 crops feed the world. Five cereal crops provide 60% of energy intake of the world population. Some 7.4 million samples of crop diversity are stored in 1,750 gene banks around the world. And uh, crop improvement accounts for 50% of the world's food. The credit how you can take it's accounted because the production agronomic way of production technology improvement also contributes for the increase in the yield. That's why so this crop improvement is improvement. I mean important. So we'll stop it here for this today's class. Factors limiting genetic improvement of croplands. We learn, then we will understand the basics. What is that problems are there in crops, how the plant breeder can tackle those things if possible. But at least knowing it is important to look into that in the next class. Thank you.